Hi, my name is Hasika Shazani bint Ismail with metric number 78022, and this is the historical context of molecular mechanism of gene expressions in human cell studies throughout the year.
My name is Renan Isabel Banus, and now I will be presenting about the gene expression overview. So, the gene expression is the process by which genetic information is translated into a functional product, mainly through RNA transcription, and this results in proteins or non-coding RNA molecules. Next is the stages of protein expression. The process involves several stages, which is firstly is the transcription, secondly is the mRNA splicing, next is the translation, and lastly is the post-translational modification. And each stage is essential for forming the final protein product. Next is the regulation of gene expression. Regulation can occur at any stages, which affecting the quantity and timing of protein produced. And this regulation is crucial for normal cellular function, adaptation, homeostasis, and recovery from damage.
Basic concept of gene expression. Exon, known as coding region of the gene that contains information included in the mRNA. Introns are non-coding region that are removed from the RNA before it is translated. Promoter, a DNA region that helps RNA polymerase to start transcribing the gene. And finally, enhancer, a non-coding sequence that help activate the expression of target genes transcribed by RNA. Central dogma is a molecular biology theory that explains the flow of genetic information. It tells that DNA undergoes transcription in the nucleus to produce RNA, and RNA leaves the nucleus and transfers the information to the ribosome, which in the end produce proteins.
Assalamualaikum and hi everyone. My name is Namika Nazara binti Sabini and my metric number is 78476. Today I will explain on transcription and RNA processing. In gene expression, the first step is transcription and occurs in the cell nucleus. Here, RNA polymerase attached to a DNA promoter and create a complementary RNA strand called pre-mRNA, which contain both intron and exons. The pre-mRNA is then processed by adding a 5' and a poly A tail for protection and export. Splicing removes the intron and join the exons, forming mature mRNA. This mature mRNA is transported to the cytoplasm for translation into proteins, decoding genetic information to create proteins in cells. Next, I will explain on translation and post-translational modification. Once mature mRNA leaves the nucleus, translation occurs in the cytoplasm, turning mRNA into a protein. Ribosome read mRNA codon, each coding for a specific amino acid. Transfer RNA molecules bring the corresponding amino acid to the ribosome, forming a polypeptide chain that folds into a functional protein. Post-translational modifications such as glycosylation and ubiquitination tweak the protein for full functionality, affecting cell signaling, protein stability, and degradation. This modification ensures proteins function correctly, regulating cellular activity and adaptation. That is all from me. Thank you.
This is the quantitative analysis of gene expression. The quantitative analysis measures gene expression levels in specific cells, tissue, or organism, and it is vital for understanding the relationship between gene expression profiles and phenotypes, and helps biomedical scientists decipher cellular function. Lastly is the application in animal biotechnology. In animal biotechnology, this analysis is essential for generating transgenic or knockout animals. Unlike simple in vitro system, animal organs exhibit greater cellular heterogeneity, making gene expression analysis and regulation more challenging but also insightful.
Applications in alternative splicing, including a human gene TTN that encodes the muscle protein titin, consisting of 364 coding exons and 4039 types of splicing processes. Other than that, is sex determination in Drosophila melanogaster by skipping off an exon due to high X to A ratio during mRNA splicing. For therapeutic applications, ASOs are used in the allele-specific strategies in Huntington's diseases, and other than that is by targeting multiple SNPs with tightly linked amplified mutations within the introns and exons regions.
Application of RNA interference in gene therapy. It offers several benefits in cancer therapy, including high specificity, cheap cost, and efficient growth control of advanced stage tumor. Because RNA interference can inhibit numerous genes of different pathways at once, it may help lower drug resistance.
So here is the novel therapeutic approach for diseases such as cancer, neurological disorder, and metabolic problems, involve the application of targeting post-translational modifications, including dysregulated kinase, protein phosphorylation, ubiquitin proteasome interactions, and histone acetylation. Next, I will pass to the next presenter.
To conclude, gene expression in human cell studies still need to overcome technical and biological limitations, especially in capturing the full complexity of gene expressions and the ethical concerns. And in the future, the study should be able to provide deeper understanding in human disease, and genome editing tools will be revolutionized and be a norm in the society.